Why does aging occur at all? And the reason why this is important to ask, it's not a philosophical question, it's not a metaphysical question, it is a fundamental question in biology. And the reason it's important to ask this is that if you understand why something has been shaped in the way it has, and things in biology are shaped through the action of natural selection, then you're in a much, much better position to ask what are the mechanisms that drive this and how might we be able to intervene in these mechanisms. Now, I think it's worth beginning by just saying, what is aging? Actually, aging is something that, although we take for granted, people have found quite difficult to define. And the difficulty remains. And if one goes to the Gerontological Society meetings, you find that time and again people come back to this question, what is aging? And the current editor of the Journal of Gerontology, one of the journals of the big American society, is actually organizing a series of think piece articles, what is aging? Well, sort of for all of you, of course you know what aging is. We all know what aging is. We learn very early in life that aging is that process that means that as the years clock by on your personal counter of life, you become more liable to fall sick and die. And from this, more years ago than I care to remember now, came a concept that I proposed in a paper in Nature back in 1977, which is called the disposable soma theory. And this says that our genomes, our genes, evolved to limit the investments they make in the maintenance and repair of the body so that we have cells that are good enough to keep us going through 40 years and maybe a little bit more, which is what we have. We have a body that maintains itself pretty well through our teens, 20s, 30s, into our 40s a little bit. And it's after that that we begin to see deterioration appearing. And for mice that have much more dangerous lives, and where in the wild a mouse, 95% of wild mice are dead before they're 1 year old. So for a mouse, all it needs is a body that keeps it in good shape for perhaps 15, 18 months. And that's what a mouse has. It's got a body that does that beautifully. If you keep a mouse in perfect conditions, it will fall apart. It will die of old age, aged 3 years. But it's got a body which will keep itself fine for 15, 18 months. That's plenty for the natural mouse. Now, the implications of this are really quite sweeping. First of all, there is no genetic program for aging. This is pretty much established now as fact. It follows from the logic of what I've told you, but, the evidence is absent for a genetic program. We age because in our evolutionary past, it would have been too expensive to build a body that might last forever. And the outcome of that logic is that aging is caused by damage. Longevity, and we know that there is variation within the population, there are genetic factors that influence longevity. To some extent, longevity does run in families. But the genetic factors that influence longevity are factors that regulate the body's capacity for maintenance and repair. In one sense, quite a simple picture. In another sense, the simplicity belies the fundamental complexity of the mechanisms that are at play. This is complicated science, and obviously I can't give you a comprehensive overview, but I would just like to give you some flavour of what we're learning about the mechanisms in what I'll say next, and what it means for those of us who are grappling clinically with the challenges of old age. Now, when we look at aging at the cellular level, of course our bodies are made up of cells. We contain typically 100 million cells. And so we can study aging at the level of the individual cells, because cells also age. And the question that we need to answer is, what is it about the aging process that renders the aged cell or the aged organ or tissue more vulnerable to pathology? We've known for 50 years that you can take human skin cells from a small biopsy, you grow them in culture, they grow and grow and grow, and a chap called Leonard Hayflick showed back in the early 1960s that they have a limit on how many cell divisions they can go through. Not an absolute limit, it's turned out. It is itself subject to the kind of random variation that I talked about, but it is effectively a limit. So cells go through this process, dividing and dividing and dividing, and then they cannot divide anymore. Interestingly, they can continue to survive and to function, sometimes for quite long periods. In this graph, I'm showing you images of cells that were dividing and dividing and dividing for 6 months. They'd divided on the average 50 times each at that point, but then they were capable of being maintained viable in culture for 2 years after they had stopped being able to divide. But the important thing to observe here is that we have the cell, within the cell, the capacity to detect that the cell is damaged and to take action accordingly. Now, in a sense, as we go through life in terms of damage, it's like sort of living within a sinking ship. The body is accumulating damage all the time. And damage can do dangerous things to cells. Among the things it can do is it can initiate a cancer. So what our cells over the course of evolution have learnt to do is, we learned some 20-odd years ago that one thing they do is that when a cell is very badly damaged, and particularly if that cell is a critical cell within sort of maybe a stem cell or a cell that has important proliferative capacity, when it gets badly damaged, the cell turns on an auto-destruct mechanism. It commits suicide, a process known as apoptosis. So we've studied that, and we've known about that for quite a long time. That's for cells that are critically dangerous for the survival of the organism if they are not taken out of the loop. But we've also discovered recently that if you play around with the genetic mechanisms that detect damage and initiate the process of apoptosis in a cell, and if you generate a transgenic mouse that is more likely to initiate apoptosis at a given level of damage than the wild-type mouse, that mouse, it does, what apoptosis is partly there for. It protects the mouse better against cancer. They get fewer cancers, but they age faster. And the reason they age faster is that they lose cells faster than the wild-type. So what we've discovered in the work that was published a couple of years ago is that for the ordinary cells of the body, the common or garden cells of the body, it's not so critical that you actually get rid of them. They can stick around, but you just don't want them ever to divide again. So for these cells, what happens is that you've got a detection mechanism that activates this kind of feedback loop that's illustrated on the diagram, and it locks that cell in a senescent state where it will continue to be a useful part of the tissue, but it will never divide again. But what we're moving towards now is a picture where the focus has tended to be on studying end-stage pathology, on studying, the Lewy bodies in the brain of someone with Parkinson's disease, the plaques and tangles with Alzheimer's disease, the fibrosis in sort of age-related deterioration of other organs. What we have to do is to move upstream in terms of the causal pathways, because that's where intervention is much more likely to be successful. And the exciting thing that's coming out as we do this is that we move upstream from one disease, for example, Alzheimer's disease, into the headwaters that initiate the damage that leads to that condition. We find that we are in the same areas that lead to other age-related conditions. So what we are learning, this takes the earlier concept that I presented to you of aging being driven by random molecular damage, building up defects, leading to frailty, disability, disease, and it says that this is a process which is intrinsic to our biology, but it is acted upon by the circumstances of our lives. So stress, adverse environments, poor nutrition actually make the damage happen faster. Healthy lifestyle, healthy nutrition slow the accumulation of damage by empowering the body's maintenance and repair processes.